Okay, can you all see the PowerPoint?
Yes.
Okay, so I made a PowerPoint so we had a visual to kind of see. Like Bill said, I'm the Orange County Chapter Social Worker for HDSA, and I primarily do the support group, which is held every second Sunday of the month. So a little background about myself. I'm originally from Monterey Bay, which is in Northern California, so I'm a NorCal native, but I actually moved down here for school many years ago. Attended Cal State Long Beach, got my bachelor's degree there in human development, and then worked in the field for a couple years at a youth shelter called Casa Youth Shelter, working with at-risk youth. And then later went on to pursue my master's degree. Got my master's in social work, attended USC, and now I currently work as a social work care manager for Providence St. Joseph Health in the ambulatory care management department. And then of course the OC chapter social worker. I'm also married and I have 2 little boys, 10 and 4. So I'm super busy, but I love what I do. So it doesn't feel like work most of the time. I'm very passionate about my work. So Orange County Chapter. Like I said, my main role is to facilitate the support group. I run this group every second Sunday of the month from 6 to 8 p.m. Pre-COVID, we were doing 2 separate groups. I co-facilitated with Daniel Ignacio, who ran the affected population, and I primarily supported our caregivers. And then on top of the group, I also have a phone number, a line that acts as a crisis hotline. So I'll assess our callers' needs, provide guidance and some brief supportive therapy. But my main role and objective is to be this resource broker, provide the resource, do some light coordination, but really hand off that person to a referral source or alternative support system, just so I'm able to meet as many needs or many members of our community and provide a lot of support to the community. Some of the main needs that our callers will have is really lack of caregiving support. Those that are affected with the disease, it's progressing, they're not able to care for their loved ones adequately. I will assess their needs over the phone and kind of understand, what level of care do they need? Do they need help with placement? Do they need help understanding different levels such as skilled nursing facility, assisted living? What are the true needs of the house? And then provide resources, whether that's caregiving resources like in-home supportive services, finding a caregiving agency, different things like that. And then for our caregivers, with the group on Sundays, one of the biggest things that they struggled with was caregiver burnout. So really trying to help educate them on what are the signs of caregiver burnout, what resources are available. And fortunately right now, telehealth is a really big thing. And so trying to bridge resources that we have in our community for them to utilize during this time, especially with the pandemic that's happening. And, of course, with COVID, rolling with the punches, we've really had to pivot what we're doing. In the beginning, I, initially thought, okay, we're going through this pandemic. It's going to be a couple weeks. We'll go back to being in person. So I was taking it very slowly, month by month. What are we doing? Are we going back in person, or where do we go from here? So we've fortunately had Zoom to have this platform to provide this support group. We had 2 split groups, but now it's made into one. Some challenges with that is that our caregivers don't have, their own individual group that they can be in. So going forward, we're going to have to try to figure out, can we have 2 separate groups? How can we meet the needs of our community? And, what is that going to look like? And then challenges, of course, technical difficulties, low engagement, trying to promote this as something that we have available. But in the last couple months with the use of social media and reaching out to the community, I've gotten a lot of new members to join our group. So while there's challenges, the silver lining is that we have this platform that's really great and that we're able to utilize. And next steps is to possibly host multiple groups where I can provide one for our caregivers and then one for our affected members. So that's a very quick update on our support group. I have started this, it's been a little over a year since I've been in this role, and 8 of those months have been providing this support group through Zoom. So it's been a lot of learn as we go and really trying to just embrace what's happening and, roll with the punches, like I put in the title, is, trying to figure out, how can I best support our community? Where can we go from here and what to do? And, clearly the pandemic is not going away. And so really trying to figure out what's the next step for 2021. Like, what are we going to do to best support our community, provide more individual outreaches, utilizing social media to gain more support from the community and let them know that we're available. So that's that.
Great, thanks, Priscilla. Question for you: could you go a little more into the HDSA telehealth and how that works?
Oh yes. So I get a lot of phone calls where our members really need that, supportive therapy. And while that's within my scope of practice as a social worker in this exact role, I really try to link them to something that's more sustainable since I'm not doing telehealth over the phone. So HDSA provides telehealth. There's a website that I will email out to them, and then a contact that they can get connected to, and it's a free service that's from HDSA. I've had a couple members who have utilized it. Another good resource is the OC Warm Handoff or Handline, which is another resource for therapy as well.
Oh, great, thanks. I'm glad to hear that, because I know that the telehealth actually existed prior to COVID and was not created as a response, because I think maybe people may be unaware that support that you provide to us in Orange County is not possible or consistent across the entirety of HDSA's area. And so there are many areas where people do not have the opportunity to go to support. And so, HDSA created that as a method by which individuals could still get, some care and support that they needed. And so I think obviously it works well in this state of, COVID, where everything's gone virtual, as, maybe an even more important resource. So I'm glad to hear that.
And with the support group too, because it's not just in Orange County, since it's virtual, I've had someone who came across our number who lived in Stockton and had actually joined our group last month. So because it's available, I mean, it's Orange County and I'm more well-versed with resources out this way. They still wanted to come join the group just to be able to have that, sense of community and support, which was good.
Yeah, fantastic. Another silver lining, as you put it.
Yeah.
Well, thank you. So really appreciate this. And a key reason why we wanted to have you here today to talk about this is because I think most of us, we go in, COVID appears, we go into this pandemic, and it feels like in most ways the world is pretty much shut down. And certainly for the chapter, it did with regards to our events and activities, but your work with us and the support you provide, support group, and all of these benefits that you have listed here have not stopped. Like you said, there was an initial reaction phase of, okay, this is going to be a short-term thing. How do we react? But even through all of the variety of extremes we suffered this year as a result of COVID upon society, is your work and our support group has been consistent, and it's still a tool available to our local HD community. And so really wanted to emphasize that, because we do a lot of things as a chapter and as an organization, but one of the things we're very proud of is the work that you do, because as I mentioned before, not every chapter or affiliate has the opportunity to provide social worker support and support groups. And so it really is a key benefit that the local HD community here has access to. And so, want to continue to encourage people to utilize it, because it is there. You are there. You're doing great work, and you've kept that consistency across the entirety of this crazy year. So thank you for that. I really appreciate you taking the time to remind folks that we're still here. You're still here, and then also to really go into what you are able to provide our folks. I know that everyone needs it, and probably this year more than any other. All right, so with that, thank you again, Priscilla. We're going to turn it over to Dr. Leora Fox. Sorry, I'll say that better. Dr. Leora Fox, excuse me. Thank you. An easy name not to butcher, and I still managed to do it.
Oh gosh, not at all. You got it. Let's share the screen. Oop. Is that right? Let's see if I can get the slideshow going. Do you see my slides?
Yes.
All righty. Hi, everybody. My name is Leora. I'm coming to you from Albuquerque, New Mexico. I was previously in the New York City offices of HDSA, but I moved to the Southwest recently with my husband and our 20-month-old daughter. I've been at HDSA since 2017, and my background is in HD research. I did my PhD at Columbia, studying HD and how the brain clears away deposits of harmful Huntingtin. And I also have some experience in science writing. I write for HD Buzz. And my current role at HDSA is the Senior Manager of Research Programs. I want to move around my screen a little bit. Sorry, give me 1 second. I feel like I'm still struggling with the technology, even though it's been long enough. All right. So my job basically includes making sure that the scientists that we fund are feeling supported and making progress in their work. And more importantly, it includes keeping up to date on what's going on in HD clinical research, so I can help get that news out to the community in different ways. So through the HD News site, HD Buzz, through HDSA's weekly research blog, through our webinars. We have a clinical trials matching service, HD Trialfinder, and we come to events like this one. So thank you very much for having me today. And I'm going to focus on this presentation is roughly 25 minutes. You can butt in at any time and be like, I didn't get that. It's a lot of cool stuff going on. So yeah, I'm going to focus on a broad view of clinical trial news. So what's in the pipeline today, what HD genetic treatments are being tested, as well as other drugs that focus on particular symptoms or other aspects of HD biology. I'll also touch on observational research like the Enroll HD study and how this is shaping the new hot topics in HD research that could become the HD drugs of the future. And finally, I will share some ways that you can stay up to date on HD research and get involved. Don't worry about taking notes. I know this is being recorded now, and I'm happy to share my slides as well. And you can always reach out to me with research questions. So when we talk about Huntington's disease clinical trials and the HD drug pipeline, what we really mean by a pipeline is everything from drug discovery, so understanding the biology of HD, discovering what chemicals can alter that and how, testing them in cells and animals, all the way through different stages of clinical trials in humans. And just so we're on the same page, I want to make sure that you're aware of what it really means when we say that a trial is in a particular phase. So a phase 1 study is a first step. It's usually about 20 to 50 individuals, sometimes healthy, sometimes with symptoms of HD. And it tests whether the drug is safe to give to humans. A phase 2 is a little larger. In the HD field, it's usually around 50 to 200 people. And a phase 2 study also is testing safety and side effects and how the drug affects the body. Also, sometimes a little bit of its effects on symptoms. But then a phase 3 study is really the final step needed to approve a drug. And that's much larger. So in HD trials, it's usually around 200 to 1,000 people. And this is really where the key goal is to see whether the drug can help with symptoms or slow down the changes that come with HD. And finally, after these trials, the drug company has to go with their results to government health agencies like the FDA in the US or the EHA in Europe to get approval to produce the drug so it can be given to people. Now, this is what the HD drug pipeline looks like in 2020, with the companies and drugs on the left and the stage that they're in on the top. And the arrows are showing you what they're up to. And this is a very busy slide. There are even more companies and drugs that I could add here, but I really wanted to use this slide to show you that there are truly dozens of companies working on HD right now. So as bad as 2020 has been, there's lots of really cool stuff going on in the HD research space this year. And many of these companies are running clinical trials, are planning them in the next few years. And the largest area of focus right now is Huntingtin lowering. So to understand the Huntingtin lowering treatments that are in the pipeline, we need to talk a little bit about genetics and Huntingtin. So our genetic code's made up of nucleotides, and we represent these with the letters A, C, T, and G. So we have tens of thousands of genes, and HD is caused by this one gene called Huntingtin. And everybody in the world has the Huntingtin gene, and everybody has multiple repeats of the letters CAG in their Huntingtin gene. The average is about 20. Most people have between around 10 and 26, and HD occurs when there are 40 or more CAG repeats. There is some variability, so sometimes it's 35 or 36 or more, but in general, we know that HD is caused by too many CAG repeats in the Huntingtin gene. So a lot of us learned in biology class in middle school or in high school that our genetic code, our DNA, is turned into an RNA message, which then becomes a protein. And proteins are really the machines that run everything in our cells. So we have the Huntingtin DNA, we have the Huntingtin RNA, which is a recipe for a Huntingtin protein, DNA, RNA, protein. And because a person usually inherits HD from only one parent, people with HD have one copy of the gene from mom and one from dad. And that means that they will have some healthy Huntingtin protein. And because of all those extra CAGs that they inherited from one parent, they also have some expanded Huntingtin. Sometimes this is known as mutant Huntingtin. And we know that Huntingtin is important during the development of the brain for developing healthy neurons. It's got different functions in the brain and body. And when it becomes expanded because of too many CAG repeats, it can form these protein clumps that sort of gum up the works in brain cells and can harm them and make them sick, which is really what leads to the symptoms of HD. So the expanded Huntingtin protein is causing cells to get sick. And the whole concept behind Huntingtin lowering is to try and get rid of some of that harmful Huntingtin by making less of it. So if you think a little bit about this like a cookbook, you have an old family cookbook full of recipes, you decide you want to make cupcakes. But there's a mistake in the cupcake recipe. There's one tiny mistake in the whole cookbook in that cupcake recipe that tells you to add a tablespoon instead of a teaspoon of baking powder. And then if you do this, you make a copy of that old family cookbook and you bring the cupcake recipe to the kitchen. Then you make that recipe and your cupcakes are really going to come out very weird because of that one little mistake. And of course, the solution to that is to make sure that you don't use that bad recipe again. So crumple it up or cross it out or remind yourself that those cupcakes exploded in the oven and didn't taste great. So the vast majority of Huntingtin lowering approaches are using this same kind of strategy. So basically doing something to interfere with the Huntingtin RNA. So crumpling up that recipe that makes the expanded protein so that there's less of it. And again, people with HD have some healthy and some expanded Huntingtin. And some researchers and companies are focusing on lowering all forms of Huntingtin, and others are focusing on lowering just the expanded form of Huntingtin. And in general, how this works is that drugs are designed to stick to a part of the Huntingtin RNA and either trick the cell into not using or destroying the recipe because it's not readable, or the drug might cause the recipe to be chopped up. But in general, the goal is to stop brain cells from making harmful Huntingtin protein. And the clinical trials that are in progress right now are testing first whether this works in humans and is safe in humans. And second, whether this can actually slow or stop the progression of HD symptoms. And here are just some of the companies that are working on Huntingtin lowering. Again, this is a very busy and jargony slide, but I'm just using it here to really emphasize that there is unprecedented activity in the pharmaceutical industry around creating the best genetic drug for HD, or different kinds of genetic drugs for HD using lots of different approaches. So this is a very exciting pipeline just for Huntingtin lowering. And there are a few studies that are already in clinical trials. There are 2 companies testing ASOs, which are genetic drugs that are delivered via spinal injections. Roche, Genentech, and Wave are using this approach. There are gene therapies. So uniQure is testing a gene therapy delivered by a one-time brain surgery. And both Novartis and PTC Therapeutics have announced recently that they'll be testing an oral drug. And the idea of a pill is kind of the holy grail of Huntingtin lowering. So I want to summarize the latest news about each of these trials. So Roche, which is known as Genentech in the US, is testing their Huntingtin lowering ASO, which is called tominersen. And it aims to lower the amount of total Huntingtin, both good and bad. And they are conducting a phase 3 trial right now. It's called Generation HD1. And this is the therapy that is really furthest along the pipeline right now. There are nearly 800 people participating at more than 100 sites in 18 countries. And I can also share that this trial recruited in absolutely record time. So Roche has shared with us that it was their fastest recruiting trial ever. And I don't mean like HD trial or rare disease trial. I mean, in the history of this big company, this is the fastest any trial had recruited. And that really speaks to the strength and the engagement of this community in research. So everybody in this trial will participate for 2 years. They receive spinal injections and undergo evaluations at study sites every 2 months. And Roche recently announced that they expect to be able to share the trial results in 2022. And this is really the test as to whether this drug can help slow down the symptoms of HD. Wave Life Sciences is conducting 2 trials of 2 ASO drugs, also with spinal delivery. And these trials are called Precision HD1 and Precision HD2. And both of these are testing mostly safety and the ability of the drug to lower harmful Huntingtin while keeping healthy Huntingtin intact. And this is really what distinguishes Wave's approach from Roche's approach. So because of the way they target just the harmful Huntingtin, both of these drugs would only work in a certain percentage of people, around 70%, who have to have a particular feature in their Huntingtin gene, a spelling difference between the 2 copies of Huntingtin. And that's how they target just the mutant Huntingtin. So late last year, Wave announced their initial results. And so far what we know is that these drugs have been safe and well tolerated and seem to be showing promise for lowering Huntingtin. But they did decide to add a higher dose group of participants, and they announced a month or 2 ago that the full results from both studies are expected in early 2021. So in the next few months, we'll know better whether these drugs are safe and whether they can lower Huntingtin. They're also planning a 3rd study, which would increase the percentage of people that could receive this kind of treatment. Now, uniQure is conducting the first ever gene therapy trial for HD. And gene therapy is the idea of actually putting genetic material into a person's cells to treat the genetic cause of a disease. ASOs, like I was speaking about with Roche and Wave, need to be delivered regularly, but uniQure's approach is a one-time treatment, and they use a virus to get it inside of brain cells. Nobody wants to hear about a virus right now, but this is a harmless one. It's very good at getting inside cells, and researchers can fill it with the right genetic material to get brain cells to basically churn out a drug, and it's delivered via a brain surgery. The drug is called AMT-130, and basically what it does is it sticks to and covers up the Huntingtin recipe so that less protein is made. So yeah, this is a small safety trial. There are going to be 26 people in this trial. And because this is kind of an unprecedented type of trial, They had 2 folks undergo the surgery in June of 2020, and then they waited 90 days to make sure that there weren't any dangerous side effects. And then the next 2 people participated in October. And if this continues to go safely, then the trial will open up to the rest of the 26 people. So it's a slow-moving safety trial, but it's a really unprecedented and exciting type of therapy. Novartis also announced recently that they're planning a trial of a drug that has the potential to lower Huntingtin when taken by mouth. And this drug is called branaplam, and it was actually developed for a disorder called spinal muscular atrophy, which is usually fatal in young children. And this is actually a really amazing science story. So in doing a big screen of a lot of chemical compounds, it was discovered that branaplam could also lower Huntingtin. And so they began testing it in HD cells and HD mice, and they got consent to test samples from the children that were taking branaplam and found that their Huntingtin levels were also lower. So Novartis very recently received orphan drug status to test branaplam in adults with HD. They're testing it now for safety in a small group of healthy adults, and they expect to launch a phase 2 trial for HD patients in the second half of next year. There's no information yet about locations or eligibility or really what the trial would look like, but the potential for an oral Huntingtin lowering drug is very exciting. And PTC Therapeutics also just announced a few weeks ago that they have similarly begun a phase 1 trial, I believe in healthy adults, and that data is going to be available in early 2021. And yeah, it's very exciting, the prospect of a pill that you could take to lower Huntingtin. Feels like science fiction. Now, Huntingtin lowering is not the only approach to researching HD treatments. There are lots of companies developing drugs that focus on other aspects of HD biology or are designed to treat particular symptoms. So I'm going to briefly summarize the ones that are recruiting now, as well as the most recent news that we have about results and plans from other trials. So a company called Neurocrine is recruiting for a study of a drug to treat chorea, movements of HD, and this is called KINECT-HD, and the drug is valbenazine, and it's already approved for a different movement disorder called tardive dyskinesia, and people with TD also experience involuntary movements. This study for HD patients is recruiting adults at 50 sites in the US and Canada for a 12-week trial. Annexon Biosciences is conducting a small phase 2 trial of their drug ANX005, and this drug targets a part of the immune system that might be overactive in the brain during HD. And the aim here is to try and preserve the connections between neurons called synapses. It's a small study with about 25 people at a few sites in the US who either have pre-symptomatic HD or early HD. It's an open label study, so everyone involved receives the drug, and the treatment period is about 10 weeks, and then there are follow-up visits for 6 months. HDSA was particularly excited about this Annexon study because we supported some of the initial basic science that led to the development of this drug. And so it was very cool to see it move into humans. A company called Prelenia is doing a large phase 3 trial of a drug called predopidine. And a few years ago, there was a phase 2 trial of predopidine, which did not meet its goal of helping with movement symptoms, but the drug was safe, and it did show some promise in helping people with HD to maintain their daily function. So this larger and longer trial is going to involve around 480 adults who will take predopidine for 18 months to see if it can help with day-to-day function when given for a longer time. We also had some news this fall about trial results from different completed studies. So Azavan Pharmaceuticals has a drug called SRX246, and they're testing it to see whether it can help people with HD who have trouble with irritability and aggression. And in October, they announced that their phase 2 trial, which was called the STARE trial, did indeed help. And so they're now designing a larger study that will focus on people with certain types of aggressive outbursts. We also heard in October that Sage Therapeutics successfully completed a very small safety trial of a drug called SAGE-718. And this is meant to combat cognitive impairments in HD, which is a difficulty that most people with HD face early on. And they're also developing, hopefully, a larger trial of this drug. Finally, unfortunately, in September, Vasinex announced that their Signal trial was not successful. So their drug papinamab did not have a significant benefit for HD patients. But we do know that the insights from this study are going to be applied to other groups of people, including Alzheimer's patients and also people with a rare form of cancer. So Enroll HD, I'm sure lots of folks are familiar with this, but we always like to plug Enroll HD. Enroll is a big worldwide observational study. There are about 20,000 participants so far. Observational just means that it follows and watches people over time. There's no drug involved, and it's open to anybody in an HD family, whether they're gene positive or negative, whether they're symptomatic or not. You don't even need to know your gene status to participate and enroll. It involves visiting a clinic once a year for an hour or two of exams and health questions and tests, sometimes giving a blood sample. There's support available for travel. And we're always harping on Enroll because it is a hugely important study. It has created an amazing, robust data set that is a big reason why there are so many companies that are working on HD today. Studies like Enroll are really allowing researchers to track people over time in large groups and understand things like, why do certain people develop symptoms earlier or later than others? And that data is kind of helping other companies to design drugs to combat this. Enroll is helping to identify ways to track HD in the brain before symptoms occur, so that we can prepare for future trials to prevent HD. All of this data is just a really, really great resource for lots of companies and researchers, and it's a wonderful study to start participating in HD research and get to know a team of researchers. So a lot of the hot topics in HD research, the stuff that scientists are really excited about, these topics can also arise from studying people through big observational studies and then kind of following up on what they learn. And I wanted to briefly mention some of these topics. I focus on clinical stuff today, but if folks have heard of these things and have questions, yeah, you can reach out or we can chat more about them. So as I mentioned, there's interest in understanding why age of onset is so variable. And one really important thing that's been discovered fairly recently is that CAG repeats in some cells can actually get longer over time. And it's believed that this can cause symptoms to arrive earlier. So there are research groups trying to figure out why this happens. There's, in fact, a company called Triplet Therapeutics that's designing a drug to stop CAG repeats from getting longer. And Triplet recently finished recruiting an observational trial called SHIELD HD to better understand this in humans. So this topic, the idea of CAG repeats lengthening, is called somatic instability, and there's a lot of activity around this in the field right now, so you're likely to hear more about it. There are also researchers working on things like correcting the HD mutation itself. So instead of focusing on that RNA, that recipe, to go into the cookbook itself, if you will, and correct it once and for all. So you may have heard of CRISPR, and some forms of CRISPR technology are being focused on HD. It's not really a medicine yet, but CRISPR is a really interesting technology that can tell us more about HD and is being explored as a treatment. There's also a really interesting area of HD research that focuses on adult stem cells, where scientists can actually take a skin sample, for example, from an adult with HD and use different chemicals to turn these cells into stem cells that can then become any cell of the body. So they can basically grow human cells in a dish to study HD, which has fewer technical and ethical issues that are posed by using embryos. And I imagine that that Leslie will be speaking about this a bit as well, since a lot of her research focuses on that. So I'm happy to answer questions about any clinical trial info and any of those topics, but I also want to leave you with some ways to get involved and stay up to date on HD research. And I have to plug HD Trialfinder. So all of the studies that I'm talking about that are recruiting are in HD Trialfinder. It's HDSA's clinical trials matching service. So this is a website and a call center that is staffed by trained navigators. We list currently recruiting HD trials in the US and parts of Canada. You can just browse it. You can enter some basic information to see what trials you might be eligible for and get contact information for those trials. In addition to big drug trials, HD Trialfinder has lists of local and observational studies. So everything from testing a new kind of brain imaging method to trying a physical therapy regimen or a different combination of medicines, to signing up for a brain donation after you pass, and there's lots more. And these studies are also important for supporting research and care. They can also help you develop a relationship with the teams and the experts that are involved in and committed to HD research. So check out HD Trialfinder. And I want to plug again some ways to stay up to date on HD research. HDBuzz.net is a site where scientists write about HD research in plain language. HDSA's newsfeed and Facebook page, you'll find updates on research. I write a weekly research blog that's kind of a roundup of what's going on in the field and at HDSA. And again, I encourage you to check out enroll-hd.org and hdtrialfinder.org. And yeah, I'll just say thank you to everybody who's here and everybody who's watching afterwards, and just emphasize that HD research is really a collaboration between the community and between researchers, and especially clinical trials. And so the better informed we all are about what's happening, the more our voices can be heard. So whether or not you or somebody that you know can participate in clinical trials, learning about them and being aware of research is really a form of advocacy. So thanks for your time and attention, and thanks for having me, Bill.
No, thank you absolutely for joining us. It truly is a great pleasure for us to get your attention. Certainly so glad to see, I mean, I was not surprised to see the reminder and the emphasis on both HD Connect and HD Trialfinder. Expected you to bring it up, but also wanted to just reinforce that the importance of getting involved. A couple of things I remember when I first heard about it. I remember Dr. George Yorling was very clear in saying that we have, I mean, we all know that there's an incredible amount of research going on by some genius individuals such as yourself, but that, you know, the cure for HD or any cures or progress for HD is not going to be done just by researchers. It has to be performed on people, and it has to be successful in people. And kind of the nuance of HDSA, we're all glad it's a rare disease. We wouldn't want more people to have it, but that also means that your base for clinical studies subjects is a lot smaller. And so really it's upon us, the HD community, to take an active role in these research and clinical studies by being part of them, because that's where any progress or cure is going to come from. So just a little reminder about that. Really good stuff. Also, I mean, I think we talked before about how this year and the craziness we've had, it's easy to think that all things have ground to a halt as we suffer through COVID-19. But it's really great to see that the research and the studies and the trials towards HD have continued to progress. I know a lot of us are waiting for a lot of exciting news. Where obviously the Roche Genentech is the one that had everyone's attention the last time we were at a physical annual convention, and so certainly some exciting stuff. Thank you.
Yeah, that's an important point. You know, there's I also want to say that, you know, it's not just going and getting stuck with a needle. It's everything from participating in surveys to, you know, helping your loved one get somewhere to participate, to to doing this observational research. And I know that there's always a question about how COVID has affected these trials, and so I wanted to bring that up and basically although there have been slight delays in some of these trials, there have even been trials that have recruited during the pandemic, which is really quite amazing. And there, there haven't been significant delays or stops in the major Huntington lowering trials. uniQure was delayed by a couple of months, but they are, they have been able to forge ahead safely with the folks who are involved.
That's great news. Yeah, I think
Thanks again. Obviously
everyone's attention's on a COVID vaccine, but I don't know, at least personally, that's still second place in my mind as far as cures that I want to see coming out of the medical community. So thank you. I have a few more questions about research in general, but it's not the Bill show. What I'll do is I'll turn it over to Dr. Leslie Thompson. I know that she's going to speak about some related topics. And then when Dr. Thompson, you're done, maybe I'll come back and I'll field a few of my oddball questions on the research. So Dr. Leonard Fox, thank you so much again. Dr. Thompson, thank you in advance.
Okay, hi. I apologize for my dog in advance if he starts barking. But yeah, so I thought today I'd just give a few research updates. So these are the people in the lab that do all the work for our group. And I'll be highlighting a few people as I go along as well. But everybody works as a big team, and they're all wonderful. Let's see if I can get this to not stick. Okay, so you heard from Leora about this sort of central dogma that you start with DNA that's made into RNA and eventually into protein. And that's going to be relevant as I move along in the talk. We'll be focusing on different aspects of this. And the central cast member that we'll be discussing is the Huntingtin protein. And with this mutation, as you heard from Leora, also you have this repeating unit. You can see this little beads on a string here in the DNA, which is the CAG-repeat, that when it becomes protein is also expanded at the protein level as a glutamine. So we call it a polyglutamine repeat disease. And so this mutation then causes many different cellular effects, many different effects in the brain. As you heard also, you typically see movement disorder, and really these devastating declines in cognition and psychiatric well-being. And there's specific regions of the brain that are primarily affected. The major area is in the striatum, and you find a particular cell type, which I'll discuss a little bit later, called a medium spiny neuron. And that cell type is very vulnerable to the presence of this mutation and causes it to become very sick and eventually die, and you have the loss of brain tissue. Also in other regions of the brain, though, we know a lot more now that many areas of the brain are affected over time, progressively, including what's called the cortex, and that I'll also mention that. But as you also heard, there's a lot of hope for therapies. And there are so many different approaches to this. It was great to hear this overview from Leora of the different companies and different approaches that people are taking to treat different aspects of the disease. And here I'm going to cover two major areas that we've been focused. We have a lot of different projects in the lab, but I thought I would pick two that are hopefully on their way to therapeutic development, but are not quite where you heard some of the company approaches, but things that we're understanding about the disease that could lead to therapeutics. So the main focus is on what you heard about this DNA repair puzzle, why these factors that maintain the DNA in its normal condition are impaired and what we might be able to do about it. And then just the ability to preserve the synapses. So these connections between the neurons that are in the brain, to maintain these connections and keep them very healthy. We have a couple different approaches for that. And this has been a big area. The thing I'll focus on first is this DNA repair concept. And these have come out of studies from the DNA of individuals with HD that had either earlier ages of onset or later ages of onset than you might have expected from their CAG repeat length. And there's been a number of different publications in the field. that have implicated all sorts of genes in this pathway that's involved in handling DNA that gets damaged. And so the idea is that when you have an insufficient amount of DNA repair, again, as Leora nicely alluded to, you have increased damage in neurons. So in this graph where you have increasing age of onset on the left here, on the left-hand side, so somebody that has it at 40, say, might have, say, 50 repeats or 45 repeats. And to shift, the general curve is shown here, where the longer your repeat length, the earlier the age of onset in general. But again, there's this range of repeat lengths and range of timing that people can end up getting the disease. But if you look across at any given, say, repeat length, let's look over here at 45, there's a big range. And there are some genes in this pathway, if they're not working correctly, can make can have an individual get the disease at an earlier age than they might have otherwise. Likewise, there's some genes that are protective and move that curve up and have an individual have symptoms later. So we're trying to really understand this. You heard about therapeutic approaches and drugs that are targeting this, and why you go from this healthy neuron then to this very sick neuron in the brain because of this phenomenon. Also, there's this concept about somatic repeat expansion. I think people have heard about that, that the repeat itself, so you say a parent passes on 40 repeats to their child, but in the brain, that repeat can change in its size. It can become longer. It can go from 40 to 50 or 60 and have different lengths in different cell types. So in most of our cells, you maintain the same repeat length. But sometimes in the brain, if you have these defective DNA repair pathways, it will cause an expansion even further beyond your mutation that you have to cause a longer repeat length. And that repeat length, the longer that repeat length, the more deleterious, more harmful that mutant Huntingtin protein is in the brain. So what we'd really like to understand is, what are these pathways that go wrong, and how might we intervene so you prevent this from happening, this aberrant additional expansion that can go on. And somatic just means that it's cells in the body. It's not when you're passing it on from generation to generation, but rather cells that are already in your body. So these mechanisms contribute to HD, and this happens at the level we talked about with decreased genome stability. So they can fragment, the DNA can be harmed in various ways, cause mutations or become unstable. And as I mentioned, now you can have this repeat expansion that occurs as well. So there's two different ways that these DNA damage repair pathways might contribute to HD. One of the things we've been studying for a very, very long time now, this is work with Joan Steffen over the years, is what's called post-translational modifications. And these are just tags that go onto proteins that help dictate what their function is, maybe if they're where they go, are in the nucleus of the cell, are they elsewhere in the cell, do they interact with particular proteins, are they involved in clearing proteins out of the cell. So there's many different types of these little, you can think of them as postage stamps or little tags that go onto proteins at various sites. One of the ones that we've been studying for many years is called SUMO, just like the sumo wrestler. And what we know now is that this particular tag that you can put on the protein is involved in DNA damage repair. So it can tag all sorts of different proteins in the cell. In addition to Huntingtin, it can tag onto what's called the Fanconi anemia complex, which is one of these modifiers that's been implicated in Huntington's disease. It can even tag something called BRCA1, which is the risk gene for breast cancer, or P53, that is very involved in cancer, in whether an individual has cancer or not, or gets cancer. So this particular little tag, the SUMO tag, is very involved in the ability of a cell to deal with DNA damage. And that's because it will tag onto a protein and have it, including these types of proteins that are involved in DNA damage, including Huntington itself. And so we're trying to understand that. Now there's another protein, and this is about as technical as we'll get into, but another protein called PIAS1, and this is called protein inhibitor of activated STAT, but we'll just call it PIAS1. It is a protein that takes these little SUMOs and sticks them onto these cellular proteins. Okay, so it tags these proteins. It also is involved in interactions with other proteins. So it is involved, for instance, in tagging Huntington, the Huntington protein, and many of these other DNA damage repair proteins that are in the cell that are very important in these processes we've been talking about. One that we've discovered very recently, and this is working with a collaborator in Texas, I'll show you a picture in a second, is this PNKP. This is a very important DNA damage protein, and it is also SUMO modified, which had not been discovered previously. We just found that recently. And that modification needs this enzyme, utilizes this enzyme PIAS1. But when you have too much of this SUMOylation, that can be bad, and it can accumulate in the brain, and it can be a negative. So something that normally happens, if it happens too much and gets caught up and just starts to accumulate, that has negative effects. So over the years, we found that if Huntington has too much SUMO on it, it starts to clump, it starts to accumulate, it doesn't get cleared from the cell appropriately. So what we did a number of years ago is start to test whether if we either increase it, PIAS1, or decrease it, what does that do to cells or to animals? And found that if we reduce it, it's very protective. It reduces some of the accumulation and aggregation of Huntington that shouldn't be going on. But what we just found recently, and this is not published yet, although hopefully soon, is that if we reduce this in an animal that resembles human HD, it has one copy of the mutant gene and it has one copy of the normal gene, full-length gene. When we reduce PIAS1 using a virus that you heard about from one of the therapeutics, reduce PIAS1 in that mouse, or we reduce PIAS1 in human stem cells. So these are stem cells that are derived from a skin sample that were reprogrammed so that they proliferate into a dish and can be made into neurons. So either way, if we reduce that in a mouse or we reduce that in a stem cell, and then we sequence all that RNA you heard about, the RNA that dictates the protein, so it's a nice snapshot of what's going on in the cell. If we do that, we found that we reduced DNA damage, we reduced the problems with some of the DNA damage repair enzymes, primarily this PNKP that I just mentioned. So PNKP starts working again. You have increased DNA damage repair, and you rescue some of the RNA dysregulation. So the RNA isn't made properly in an HD cell, that becomes rescued with this treatment of decreasing PIAS1. And in addition to this, so this improves these synapses. The transcriptional dysregulation is largely along these to preserve these connections between the neurons. So you rescue that along with rescuing some of the DNA damage repair problems. And along the lines of that somatic repeat expansion that I talked about earlier, if you reduce PIAS1 in a mouse model, you shift that propensity to expand that repeat. So this is just a small change in this particular mouse, but you decrease the propensity to have that additional expansion of the CAG repeat in those mice when you reduce PIAS1. So you're not only fixing some of the problems with the enzymes that are involved in DNA damage repair, but you're also preventing some of the actual damage from occurring. So we're pursuing this this area of research now currently, and thinking about ways that we could clinically develop this type of approach, working with Ionis and other companies to develop ways to knock this down. So there's a lot of areas of research that are focused on these kinds of things. So the fact that you have these little bits of these areas in the nucleus that are subject to DNA damage. There's lots of other types of consequences of things like DNA damage or stress that goes on within a cell and ultimately impacts the ability to have these connections. There's things called RNA granules, which we found are increased in Huntington's disease, or stress granules, things called P-body. So there's all sorts of things that these little dots, these that are normally protective in the cell that respond to, say, a virus coming into the brain or anything. So they respond to various stresses that happen in daily life and with aging. But with Huntington's disease, with the presence of that mutation, they do not behave correctly, just like the DNA damage response. So that's a very active area of research in our lab and many other labs around the world. So that's sort of one story that's going on. The second one has to do with stem cell transplantation. I know there's a lot of interest in that area. So if you think of neurodegeneration as the loss of these structures and function of neurons, including the death of neurons. So in healthy neurons, you have these synapses I talked about, the connections between two different neurons or two different brain cell types, and they communicate with each other through electrical signals. And all of this is happening in the healthy brain. When you have HD or some other type of injury, you then have a damage to these connections between the brains, the synapses shrink up, you have broken electrical signals, and things are not properly working. So what we're trying to do with stem cells transplants, and originally we had hoped just to be able to secrete factors that would maintain a healthy environment and try to maintain the health of these neurons as well as possible. But what we're finding now is actually it's not only helping the existing neurons, it's allowing for the growth of new neurons that are forming new connections in the brain. So it's helping in two different ways. And I'll go into this a little bit now. We're using what are called neural stem cells. And these are cells that can develop into multiple types of brain cells. So they can become what are called astrocytes, oligodendrocytes, neurons, all sorts of different cells that make up the human brain. This is what they look like. They're partially differentiated, or they're partially guided along a neuronal lineage. And they look like this. They're called Nestin positive, but they're these just beautiful cultures of neural precursors that are implanted into the brain of HD mice in this case. So we've done about 3 or 4 different trials in different mouse models where we inject into the striatum of the mouse. We let it go over time. We do behavioral analysis. So we test if those mice can still perform specific tasks. HD mice are impaired in their ability. And then we look at the brain tissue and analyze what's going on deep in the brain. And this is just an example from that same mouse I showed you before that has the one good copy of Huntington's and one mutant copy. And one of the tests we do with these mice is called a running wheel, where they just run along a wheel. It's in its cage, and they tend to run a lot at night. And what we had found is, and others have found, is that when you have HD mouse, you can see this flat line on the bottom, it's really not running very much, and it's not able to run very much over time. A normal mouse, unaffected mouse, is this top line way up here. And so it is very, very active on this running wheel. When we transplant in those stem cells, you see something very much more comparable to the unaffected mouse than to the mutant mouse. So that was the first piece of good news. And we've seen this over and over again in many different mouse models where we've improved what's called the behavior of those mice. We were also surprised to see, so this particular mouse lives quite a long time, and so we're able to test transplantation over a long period of time. So we implant those cells, put them into the brain, and then we let it go for 8 months. And at the end of 8 months, we'll take a look at those brain tissues. And what we found, to our surprise, is actually many of these cells, so these green cells you can see, they turn into neurons. So we didn't know that that was going to be the case. We didn't know if they would survive. We didn't know that they could continue to become neurons in the brain. And so that was a really exciting and important piece of news for us. And then some of these even become, remember I mentioned that the medium spiny neurons are the ones that are most affected in HD. Some of these cells, when they're transplanted, become those type of cells. Not a big proportion of them, but at least some of them, and they start to engraft or make connections with other cells in the brain. So these are just stains that are showing some of this big swath of red cells here are these medium spiny neurons, and you can see the red also in this image here. So finally, we wanted to see if these connections form. So again, I mentioned that there's electrical connections between different neurons. This is called a synapse right here, this little illustration. You can see these little molecules going from one neuron to the other, as part of the synapse. And using this extremely complex, large, and expensive piece of equipment, Charles Meschel, who's a collaborator of ours from OHSU, looked at this using what's called electron microscopy. So this is a big, huge microscope that can get a glimpse of the cell at a very deep level. And what we find is there are synapses, this little, you can see in this box, and it's got an arrow to it, that forms between a mouse cell that's in the mouse brain and these human cells. There's a synapse here, and there's a synapse up here. So human to human cells and human to mouse cells. So it suggests that there are actual connections between these transplanted cells that have differentiated into different neuronal subtypes. And what we find in work with Mike Levine at UCLA is that they have they produce electrical signals, and they rescue some of these deficits of that electrical connection between the neurons. So it's been a very exciting set of findings. We have a lot of work to do still. And some of the challenges in taking this to the next step, which is to clinical trials, we're hoping to file an IND sometime in 2021, which is an investigational new drug with the FDA, to even start a phase 1 trial that you heard about from Leora. There's a lot of challenges in doing this. It's very exciting findings in the mice, but we want to make sure that it's very, very safe, that it has all the readouts we need it to have. So we formed an organization called Stem Cells for HD. It's a consortium that's worldwide. We have members in the UK and Italy and Spain, Korea, all over the world, US, to address some of these challenges and come up with the best ways to approach them so we can take this to a clinical trial. So including what are the best models, what are the readouts we should have in animals, what are the requirements to manufacture these cells so you can put them into people, and things like, because this is a human cell from someone else, you're going to have immunosuppression to be able to tolerate. It's like any other transplant. You have to have some immunosuppression so you don't reject it. And how to surgically deliver it and things, and all the regulatory phases. But we're all working very closely on this, and things are moving along very quickly, actually, on many different fronts. So we have these neural stem cells. Other individuals are using more astrocyte-like ones, or are further guided down the pathway to become a medium spiny neuron before they're transplanted. So there's many different approaches to do this, and it's I think it's going to be a reality in the not too near future for Huntington's disease. And it has a lot of possibility to both protect existing cells and to prevent loss of the circuitry of these connections that happen in the brain. And this is very dependent, and I'm going to be working closely with Enroll-HD as well for individuals with HD. And just for the second part, this is a very large group that's been working on the approach that I just showed you with the human neural stem cells from UCLA, UC Davis, that the GMP facility there that generates the cells. We have all sorts of advisors, work closely with CERN, who's been the funding agency. These are the individuals at UCI. Jack Riedling has been running this project. Surgeons, neurologists, now Anna Morenkova is working on this, and all sorts of consultants that help us as we're trying to design the clinical trials to move forward. Vicki Wheelock, Sarah, Blair, many people that everyone here is very familiar with. So thank you very much, and I'll take any questions.
So yeah, I've got a whole bunch of questions. I guess the first one on my mind is when you injected that mouse with this recipe of stem cells, and then you wait 8 months before you see, you can actually look back at the brain and see the progress. How loud is the cheer when you see all of these medium spiny neurons that have actually grown, that were beyond what you probably expected to?
Oh, it was pretty incredible. I mean, it wasn't an immediate cheer, but it was a cheer even when we saw the behavior. And we had looked, in R62 in a short-term mouse, a very rapidly progressing mouse, and had seen that they were starting to move down that pathway. But then it was, in these cells, and then especially after seeing what Charles showed us with the electron microscopy, or Mike showed with electrophysiology, or seeing, we just found those medium spiny neurons recently, actually, and that was, we were very surprised.
Yeah, because that was another thing. I mean, you're kind enough to speak at nearly every event we have, and so it's always it's just mind blowing to me how fast, or I should say, how we're always learning something new. Research tends to be thought of as more this long, process, and certainly, you hear, Dr. Fox and then other in the clinical studies will say, these things take a while to happen, but even now you're showing stuff today, and I stay, very close to the research. I find it fascinating, but things that you said, hey, we just found this recently, it's so rewarding and encouraging to see how we're just still learning more and more new stuff, and it's taking us new pathways, new avenues. That's the first time I've seen a medium spiny neuron. I feel like I'm an expert just from what you've told me through things in the past, but to actually see it and go, oh my God, there it really is, and look at that, and thinking about the concept of performing transplants and seeing these things almost magically integrate themselves into the host is just phenomenal.
No, we're very excited about it. And with the pious, I know that's a little more sort of deeper in the weeds type of concept, but it was within this last year that we realized that some of the things that we were seeing as a beneficial consequence of pious 1 reduction actually integrated with this whole concept of DNA damage repair. That was kind of, that was.
Yeah, and I've also heard you speak in the past, about the the process of how the cells go through, and I'm going to probably say the word wrong, but the autography.
Autophagy, good.
Well, I listen once in a while.
I didn't mention that, but absolutely. So Sumo is very involved in autophagy. That's when I was mentioning clearance.
Yes.
That's the mechanism. Jonas.
Yeah, and that's actually what it was. My observations in the past, you've mentioned that process at kind of a higher level, and now it almost seems like we're just learning more about these details within that process that just sheds even more light. So I don't know, it's fascinating and very encouraging.
Yeah.
One of the questions I had, and I know you touched on it a little bit, was you mentioned that it's kind of the path from the research into the clinical process. I know you had mentioned that group had formed to kind of address many of the challenges, but could you go a little more, if you don't mind, into what do cause? I mean, it's easy to get excited. Oh my God, we found this great stuff. How does that then get into the pipeline that Leora showed us?
Sure. So I'll just give you an example from where we are, but I don't know, people a lot of people have heard of Ann Rosser, I'm sure, and Steve Dennett. They are the gurus of transplantation. It started with the fetal tissue ideas and stuff, but now that we have these other types of stem cells, we can use those instead. But for our particular project, we're at the stage of what are called pivotal safety studies. So this is way before what Leora was talking about with the companies. We're at the stage where we need to make sure, so stem cells are they grow in a dish, right? They have, and you differentiate them, but we have to make sure that there's no way they're gonna form a tumor, right, in the brain. We don't wanna do anything bad. And so we're making sure that they don't cause any harm to the mice, they don't cause any kind of tumors anywhere. I mean, so far everything looks really good. And this is done by a company that's called a Good Laboratory Practice Company. So it's a very expensive and very long process of treating mice with this and then taking the mice at different time points to make sure that their livers are good, that their brain, that the tissue, everything looks good. And so that's what's called the pivotal safety studies that are going on right now. Those were going to be done, actually they would have been done by now, but because of COVID, they won't be done till next summer. And then at the same time, we're working through the manufacturing piece. We have a manufactured product that can go now into human clinical trials. We've met with the FDA for what's called a pre-IND meeting, and they've told us the things we need to have in place before we go into humans. And so we're further characterizing those cells. FDA had certain additional questions that, okay, make sure that they can't become a heart cell, make sure they can't, become another type of cell, for instance. So we're working on all those things. We had a recent what was called a shipping study. So we have to make sure that the formulation that we put these in can be sent to an OR and can be stable enough for them not to have to grow it in a hood, for instance, like we would in the lab, that there's a formulation that can be taken, the surgeon can take right out of the bottle and inject, right? So there's things like that. And those are all ongoing right now. So if all goes well, that would be sometime next summer, file the IND with the FDA, have those discussions in the fall. And then we would get to that phase 1, like one patient at a time to start and make sure that it's very safe. That's the first thing before we go any further than that. So these things do take a long time.
Yeah, but I would say, if you're talking about next year, I would say in the context of research and clinical studies, that's probably not as long a time as I've seen on many other studies. So I still look at that as great news. It's crazy to think.
If all goes well, right? You never know. But the good news too for us for HD is that CIRM was funded again. So there's the actual possibility to keep it going because they've funded all this work.
Yes, and that was going to be another question I asked is with CIRM being funded again, I assume you're talking through the approval of Prop 14 for those here in California. How does that directly benefit either the work that you're doing or the work that's happening across the state in this regard?
Oh, I can't even begin to express how important it is. It's absolutely essential because, for instance, we would not be able to get this funded by the NIH, this particular study, and at least right at the moment. And so it's essential, and they're very expensive. This next stage of clinical trials are very expensive. We'll probably partner with a company on that. But at every level for ALS, for Huntington's, for Alzheimer's, for diabetes, for retinal diseases, for everything, CIRM has been instrumental in the state of California. And we're hoping to apply for additional funding if our pivotal safety studies all go well, to take it to the next stage for clinical trials.
Fantastic.
Yep.
Yeah, no, obviously those of us in the HD community, we were all looking at that with bated breath, and so it was very encouraging to see that it passed, and
it was a huge relief.
Great job to us Californians. So going back to the topic of transplants, if there's an intelligence gene and you can transplant that, I'll be the first in line give me a few trillion cells, and then maybe I can apply to maybe sweep the floors in your lab.
No, all of us would take that one, right?
No, it really is. It's amazing. I said it before when Laura was speaking. How I personally am always blown away. It's easy to think of the research community and the clinical trial community of the royal them that when you find research discoveries and clinical study discoveries that it's done by magic, but when you see that it's real people that are doing this work, and in some cases people I even know. I was kind of smiling as you were showing your slides, saying, "Hey, I know them," and you realize that it's people in your lab, and people that are directly related to the HD community that are doing this great work. It's just mind blowing to me that it is happening. It's really remembering that it's not going to be some magic pill that comes out of some nameless, faceless entity. It's people that are working right here. So thank you so much. It's
well, and including Leora. I mean, her work on Alfie, one of these modifiers, is very exciting.
It really is.
Researchers are real people. I haven't been in a laboratory in a long time, but I'm continuously in awe of the researchers, but also glad that they're as warm and willing to share their time as Leslie. Yes, I see, Bill, that there is a question in the chat. And I don't know if Leslie has thoughts on that. The question is, will any of this research be accelerated by Project Warp Speed? And we actually just had this question in an Ask the Scientist session that HDSA did. So I thought I'd just comment on that briefly. And the answer is that while Project Warp Speed has specifically funded COVID research to promote, to push vaccines, the way that the global virology community came together to just present new techniques and create these new vaccines has really created lots of not only new science that could potentially be applied to the HD field and many other fields, but also kind of new structures for doing science. And so it's an indirect, it's very indirect, but certainly I would hope that this country and the world sees the importance of funding and promoting and collaborating on research in this same way that should help all disease communities, including rare disease communities like HD.
Yeah, good question. Thanks, Crystal. I've been one of the luxuries of being a facilitator is that I can press unmute and ask questions however I like, and you all know my passion for learning the side of what we do. So I appreciate that you are. I'm hoping I'm asking questions that maybe those in the audience or viewing the recording later may have interest in. But yeah, no, I'm glad you brought that up, because I had another question that goes through my mind. Is I know that as those of us that are familiar with this, read about the COVID progress. I guess one question I had is, they've talked about how the COVID vaccine, one of the COVID vaccines is utilizing mRNA for, I think it's a new science within that. And so I didn't know if that was something that we are looking at. Those of us on the HD research side had said, oh, interesting. Are there ways to apply that to what's happening within the HD research world?
I'll let Leora address first, and then I can.
No, you go ahead, Leslie.
Yeah, but of course, they're blowing in the garden outside. But so, not yet that I know of, although that could, I wouldn't be surprised if there's people working on that to develop almost a Huntington vaccine, although we have to be very careful because we have Huntington in all of our cells, and it's very, very important. So it would be a little different than using an mRNA to generate an antibody like the vaccine in this case, because for COVID, we don't have coronavirus in our body, so you can target that, right? But there may be aspects of this that could be feasible, and I would guess that people will be doing that. But one thing I did want to note, it's kind of a fun fact, is that so for the Moderna drug, the difference between the Moderna and Pfizer in terms of their ability one of the differences. In terms of the ability to store it differently, the -100 versus not needing that, was actually a technological development that came out of stem cell biology at Stanford. So when a person was in Irv Weisman's lab working on reprogramming stem cells, there was an aspect of what they were doing that Moderna, actually he helped found the company that Moderna is now using that helps with the mRNA vaccine. So it's kind of interesting how these fields, as Leora was saying, the things we learn and the cross-fertilization between fields and the different approaches, you just never know when they're going to jump into the HD field and help us, just like we wouldn't have necessarily thought DNA damage repair, for instance. I mean, that was this field in bacteria and in, you know, other yeast and things that people studied, and or in cancer, you know, but we didn't necessarily expect that 10 years ago.
I don't have anything better to add to that answer, but I do have a question for Leslie, actually. It's a question that we get a lot about some of the DNA repair and somatic expansion work, which is really cool stuff. And that is, often the question is, okay, well, I'm 55 years old and I am, I'm not experiencing any symptoms. I got tested when I was 40 years old and I had 42 repeats in my CAG, in my Huntington gene. If I got tested again today, would I have, if I got another blood test, would I have more CAG repeats?
You'd still have 42 repeats. Yeah, in general, that, you know, there's always the exception to the rule, but in general, you wouldn't really see the somatic repeated expansion in blood. That's constantly being renewed. The thing in the brain is that those cells are there. They're formed. They're not. There's some renewal, but they're fully differentiated into neurons, and this is where you have this repeat expansion. So you wouldn't see it in a blood cell. They're trying to figure out ways to tell what the likelihood is. I mean, there's all sorts of ways they're trying to use blood or CSF to be able to predict or guide treatments or understand what's going on in the brain, but it's difficult because of that.
Thanks for clarifying that. I know it's something that folks always ask about.
Yes, thank you. Thank you both. I mean, so fascinating. I've only been involved with HDSA for, I want to say, about 5 or so years now. And I still remember coming in, and I think at the time there was 3 or 4 studies happening and not as much research. And now, even within that short time, seeing such a proliferation of studies and seeing, and to me, what's also encouraging is it's such, like, everyone's coming at the nut from so many different directions. It's not a bunch of studies going after the same thing. It's such a wide variety that you feel, hey, if one thing is less successful, there's so many other opportunities out there that might have more success. And just to see this continual pathway, and again, you know, seeing, Leslie, what you showed us is that there's even more emerging, you know, approaches that haven't even gotten to the clinical phase yet that would, you know, be coming in ultimately at the bottom of Leora's chart, and just adding to that nonstop pipeline of research. And so it's
and I
really
should
good
say,
stuff.
I showed 2 stories. Our lab has 5 more that I didn't talk about, and other labs have other stories that are not discussed. You know, that was 2 little snapshots, right? And so the research is incredibly promising all over the world that's going on for Huntington's right now, and more researchers are being brought into the field and studying this. So it's an incredible burst of scientific activity and clinical activity that's going on. And then the organizations like HDSA, I see Ken on there, HD Care, you know, the things, the ability to be able to, you know, as part of the Coalition for the Cure, for instance, be able to jumpstart certain research areas with unrestricted funds, right? And the ability to train students. So postdoc in the lab was a Berman Topper. We've had HDSA biology fellows in the lab those kinds of things are invaluable to be able to continue this research. So these communities, I mean, we're as inspired by all of the families as anything, right? That's what keeps everybody going every day. And, you know, everyone, the HD families are so courageous and amazing and inspirational.
I think I can speak on behalf of many of them to say that I think it's like looking into a mirror. There's just as much inspiration. It's knowing that we're seeing all the good work happening, you know, both research and clinically, that, you know, kind of keeps providing that hope. So thank you so much for that. I know that we're kind of getting towards the end of our time. I really want to thank you guys again. I know that Priscilla had to depart ahead of time. She had a small window to come present. But really wanted to remind the audience, both here and those that watch this recording later, that, yeah, even through this crazy year and our organization's inability to conduct many events in person, that our support group and our support system is alive and well and functioning as well as ever. So do not hesitate to join. We have, as you heard earlier, our local support that Priscilla helps provide and facilitate, and there's also resources available through HDSA that she can connect you to, as well as, you know, local clinical resource and referrals. Certainly having Dr. Fox here, thank you so much. I probably didn't emphasize enough having the head research person within HDSA National is a huge benefit to us. I know you're a busy person, and if all the chapters and affiliates pinged you like this, you'd have no time to sleep. But
I'm certainly not in charge, but I'm happy to be here.
Well, look, we know between you and George, we'll pick you.
Yeah, they're
Just
both
don't tell I said that.
awesome. We are so grateful for Laura and George.
Yes, Laura and George, phenomenal, and so much great work. Heard you speak at convention, and obviously have known Dr. George Yurley for a while, and the passion you have for the cause and everything you do is just amazing. Like I said, you know, you're a busy person. You have your own family. You have your paid job with HDSA, and you have 52 other chapters and affiliates probably clamoring for your time. So giving it to us today, couldn't thank you enough. And obviously,
Thanks for having me.
Dr. Thompson, you're no stranger to anybody within the local community. Didn't mention that you are one of the members of the HDSA's chair of trustees, a board of trustees, which is phenomenal, helping charter the path of our good organization. You've been a pillar in the HD research community for forever and ever. And I think people still aren't quite aware of your amazingness and standing within the HD community. And I've had the luck of going to your lab. You've been very accommodating, and it's every time I go in there, I can't believe, number one, how much amazing work's coming out of there, and number two, how young and just phenomenally smart your researchers are.
Yeah, they keep getting younger.
It's very depressing. I look at my life and I said, what have I done with myself? But still, they're just such, like I said, you showed your group.
Actually,
so I'm like,
I
yeah,
noticed,
I know them.
not to embarrass him, but I noticed Jack's on the thing, and I don't know if he can show his camera for 1 second. He's probably going to kill me, but he is the one that's leading that stem cell transplantation project.
Oh, fantastic. Jack, well done.
There he is.
I mean, it's like science fiction when you think of just the concept of that. To see it really happening is unbelievable.
Yeah, we were amazed. Like you said, you touched on some of the really amazing things like medium spiny neurons and the connectivity of mouse cells to host cell to the human cells was just mind blowing. And Leslie mentioned, this is really inspirational work, and the community really drives us to work harder. So thanks to the community for that.
No, thanks to you. God, the work is so good. All right, well, so with that, I really want to thank everyone else again. As I said, we've had precious few opportunities to reach out to the local HD community. I know that our audience on this call is not huge, but I know it's also everyone's schedules are pretty compact. So we're gonna make sure this is available to everybody. But guys, really, thank you so much for your time. Thank you so much for the information. I hope that we have an opportunity to do something like this again in person. If not one of these, certainly something, even if we're just throwing some bowling balls or some pins down the lanes. We'll get face to face here pretty soon, hopefully. But until then, everyone stay healthy. Keep your family close, and stay safe.
Thanks a lot, Bill. Thanks for organizing.
All right, everyone. Thank you. Have a great day.